Okay, is anyone? Peter, are you? Who's Peter? Peter. Do you want me to round them up, Brad, or will you? Have we got a chair? Yeah, okay. Hi, how's Lucy?
Huh?
How's Lucy?
Oh, she's
Okay.
doing
She's okay.
All right, we're going to finish up
Sorry,
our
I'll just, yeah.
STAR presentation here for our first week. I guess maybe after she talks, we'll discuss a bit more what happens at 2 o'clock. There'll be some question and answer discussion period. We'll get back to you on that.
Okay, thank you. All right, welcome back for round 3. Before I start, I just wanted to say thank you so I don't forget at the end, and in particular, the pictures of people who are in my group at the moment, and it's only because they're there that any of this work gets done. I mean, a lot of what I say is certainly not mine, but the bits that are due to these guys and other people in the past, theorists and experimentalists. So what I want to do today is first of all see what happens when we take an active system which does active turbulence and put it in a channel so that it's confined. And of course, that's something you might want to do because this thing drives itself. So you might want to use it for microfluidics. Of course, if it's doing active turbulence, this is really bad because it's not going to go anywhere. But maybe you can do better than that. The reason you can't actually use it for microfluidics is that it's very, very difficult to make enough of it, of the experimental systems, for example, enough of the microtubule motor mixtures to actually do anything beyond nice little experiments in the lab. But it's still interesting to see what it does when you put it in a channel. So let me start by talking about the sequence of dynamical states, and then I'll talk about one particular dynamical state, which is rather cute, which we've called the Cayley dance state. And then I'll talk about the transition to active turbulence in a channel, because that's going to be relevant to what you're going to hear about next week or the week after from Nigel Goldenfeld. And at least one person in the audience is much more expert than me on this, so please feel free to chip in. All right, so active pneumatic, right? It's in a channel, a long, thin channel. It's in confinement. Basically, if you put it in a circle, the same sort of things apply. This is just a picture of what happens as I increase the activity. The very low activity, it doesn't do anything. It then does laminar flow along the channel. The colors are vorticity and are pretty boring for laminar flow. And then it starts waving around a bit. You get unsteady laminar flow. Then you get this state, which is an array of vortices, plus and minus vortices next to each other. The thing forms a regular pattern of vortices, and then you get active turbulence. The control parameter is the activity relative to the size of the channel. Let me, rats. Okay. What's it done? Slideshow. And I got a movie there just to show a bit more. Regularly, this is the vortex state, and that's the active turbulent state. Yes, I mean, that will come out as I go through. Yeah, I mean, this is active turbulence. Here, the active turbulence is suppressed in some way, and we have to worry about what happens to the topological defects. For some reason, that slide's gone away, but never mind, there's a blank one anyway. Okay, so what's the control parameter? Well, you have two length scales in this problem. One is the width of the channel, and the other one is essentially the size of a vortex in active turbulence, the active length scale. H is the height of my channel, the width of the channel, and the vortex size, the length scale of active turbulence, is the square root of K, the elastic constant, divided by the activity. So that thing, which I'm going to call the activity number, A, is my control parameter. And as A increases, I go from no flow to laminar flow to these small oscillations to this strange dancing state to active turbulence. And what I'm going to concentrate on first is this dancing state, because it's cute, basically. And then I'm going to look at the transition from the dancing state to active turbulence. Okay, so this dancing state is an array of vortices, which you see in many different systems. I mean, if you actually just looked at it, it would look a bit like Rayleigh-Bernard convection. And what's interesting here is what the defects do. The -1/2 defects sit at the walls, and the +1/2 defects move down the channel, half of them going this way around the vortices, and half of them going that way around the vortices. Let me show you that in a movie. If you follow one of these green dots, you'll see it's going like this. It's being carried by the flow along the channel. This is a picture of the trajectories of those +1/2 defects. The +1/2 defects, half of them move like this, and the other half move like that. And you get this amazingly regular state minus the half defects sit at the walls. So is that real? Yeah. Okay.
I was just wondering that they don't stay at one vortex. Then I saw also the vortices, they change in size.
Vortices change in size, that's right. And so you do get them crossing over. We've tried very hard to just do this as a sort of write down the equations of motion just for the defects, but we can never get something which carries on for long enough in the simulations. We just haven't got it accurate enough that we actually can reproduce that. Okay, this is the same thing again, which probably is a movie where you can see more easily those defects moving. And so those were simulations. They were really quite well-organized simulations with very careful parallel boundary conditions and making sure that you had the right number of defects in this system. We thought we were theorists playing around. And so we were interested when in the group in Barcelona, in Francesc Sagre's group, they wanted to look at experiments on this sort of system. So these are these microtubule motor protein experiments. The only difference is that we take the stuff and we confine it. And they did this in a clever way. Here's an interface with the microtubule motor system sitting on the interface. So this is this active fluid on the interface. And then they had a mask which looked like this, which they just gently put down on the interface. And the reason that that was a good thing to do is that then the system was connected underneath these different channels. So you had the same active fluid in each of the channel, and it was easy to compare the different channel widths to see the different states that you would get. So this is a sideways view. Here's a wide channel. Here's a narrow channel. And you might ask what happens.
What's the length scale?
The length scale is, this is the width, and this is in microns, yeah. So this is the width of the channel, and they're long compared to the width. And they're not quite an open channel to make a difference in a minute that they're actually a closed channel. So in an intermediate width, what they immediately saw was this. And those are the defects, those are the plus or half defects. There they are doing this Cayley dance. The reason I called it a Cayley dance, you'll know if you do square dancing, right? It's like a grand chain. These things are moving past each other like this. It was probably a mistake because I always have to explain why I did that, but there we are. Okay, so you're sort of seeing something pretty much like in the simulations, but it's not quite like in the simulations, right? Because these things don't go on forever. And there are two reasons for that. One is noise. Experiments, are real things that any sort of fluctuation is going to disturb the position of those defects. And the second one is the boundary conditions. In the simulations, we had fixed boundary conditions, so the boundaries are absolutely tied down. That's not what happens in these real systems. And the way to see that is to look at a slightly narrower channel where you would expect Poussin flow, just laminar flow along the channel. Although remember here, I've got my channel ends closed, so you don't get laminar flow. What you might expect to get is shear flow along this channel going one direction, coming back again. Okay, and these are the experimental results for these shear flows. And what you see is a very nice shear flow there. But then what happens is you get these periodic bursts of defects. Those are pairs of defects being created. Then the thing goes back to shear flow, and then you get the defects being created and so on. And you get a almost periodic creation of the defects. And the reason for this is the boundary conditions. In a shear flow, the microtubules get stretched along these boundaries. Now, we know that if things are stretched out so they look pneumatic, this is active turbulent-like stuff, and so that's an unstable state. It's unstable to the creation of defects, to one of these instabilities and the creation of defects. So what's happening is that the shear is stretching things out. There's an instability, the defects form, which is equivalent to the moving into the channel, and then they move essentially across to the other side, and they annihilate on the -1/2 defects on the other side. Yeah. Mm-hmm. It would depend on, I mean, no one's actually done it. Yes, I would expect this effect, but it would depend a bit on how active it was. If it wasn't very active, it probably would just be laminar flow. But then as soon as it can pull the edges of the channel, pull the microtubules on the edges of the channel so they're stretched out, then you get this instability. And note, there's not a no-slip condition. There's no slip condition on the underlying sort of water around this thing, but the microtubules themselves can slip over the surface. So that's a bit different from standard pulsar flow. Yeah. I haven't properly answered the question about the instability versus the width of the channel that Leo was asking. Okay, you can think of it like that. This thing here would like to undergo an instability, but the instability is cut off by the channel because the wavelength of the instability is larger than that of the channel. It's trying hard to make the instability here. Once the instability can form, what happens is that you go into this state. Once you can fit more than 2 wavelengths of the instability with a big regal sign, then you end up in the turbulent state. So you can sort of see how you go through those different stages. Okay, so that's this interesting dancing state. Yeah. In our simulations, it was no slip. In the real experiments, it's essentially a slip condition, stress-free condition. Yeah.
Omega is what again?
That would be omega's the vorticity. So the color scale is the vorticity, which is silly really in those plots. But here you can see you have, shows you the rolls that are going on there. Yep. Surface, the channel is a solid wall, so surface tension isn't really the right thing to say, right? What you're really saying is just the microtubules can move past that wall. I mean, the interactions are small between the microtubules and the wall, let's say it like that, yeah.
How do you enforce positive vorticity?
Does it itself.
Okay.
It's just a channel. This is active, it's active. So it itself is being active. So you're not pushing it, you're not putting any forces on it. It just self-organizes like this.
Opposite shears, I guess.
Well, I guess it's a shear which goes like this in that case. But yes, here there's going to be the reason I'm umming and ahhing is that the theory tends to put no slip boundary conditions, okay? And then in terms of if I have no slip boundary conditions, right, you get a profile like that. It's driven by the fact that across the system, the liquid crystal nematogens are organized like this. Therefore, you've got a bend in the order parameter, then you've got this active stress. That's what's happening. You're getting a pneumatic order parameter which has bends in it, which gives you active stress, which gives you flow. but because the instability is cut off, you get regular flow and not active turbulence. Yeah, yeah. Well, it sets the shear rate, right? I'm not doing anything to this except sitting there and putting this active fluid in there. Right? I'm not doing that. It's just a channel sitting there. It's active, it's making its own energy. It's taking energy from its surroundings and using it to do work. Yeah. Yeah. This is essentially 1D in the spanwise direction. Remember, it was a layer, and then that layer you cut off with your boundary. It doesn't make much difference if it's 2D. There's some subtleties, but you still get this set of these states which are laminar and then go to this dancing state and then go to active turbulence. No, no, sorry. It's if you looked at it, you get these alternate vortices, but the theory is totally different. Can't remember, sorry. I mean, someone has looked at the structure of what's going on here, okay? The structure in phase space, and you can certainly see a crossover between different fixed points in phase space, and I can't remember if it was a jump or not, sorry. Yeah. Okay, so then now I want to look at the transition between this state, this regular array of vortices, and active turbulence. Between the regular array of vortices and active turbulence, this is a much longer bit of channel, and it shows you how the transition works. What happens is that you get regions where the vorticity is nice and ordered. So this is the active turbulent, this dancing state. And then you get sort of localized regions where everything goes wrong and you get active turbulence. So nice ordered regions, regions which have active turbulence, nice ordered regions. And you can work out where these bits are by measuring the vorticity relative to the vorticity of this nice ordered state with the ordered vortices, and in particular measuring that vorticity squared, the Enstrophy. But it doesn't matter quite what I measure, okay? These bits show up, and these are the bits where I want to follow how they evolve with time. Yeah. I don't think well, as far as I know, the defects are not terribly important in this. Here the defects are doing nice regular things, and here the defects are much more random, but that doesn't seem to be implicated in what's going on. It's just as you pour in more activity, you end up with these states appearing. Yeah.
Does this pattern jump around in space? This is a snapshot.
This is a snapshot, and what I'm going to do now is show you a kymograph, which is how this evolves with time. A kymograph, yeah, that's the next slide. A kymograph is the time evolution of this. So what I care about is where these patches are as a function of time. Okay. And if you look at where the patches are as a function of time, right? So this is one of these little patches. And then this is time. What happens is that they can split and they can disappear. And if you're well and truly in the state where you get just the vortex rolls, they all die out and they disappear. Here, if you get a little patch, what I'm doing now is taking this activity number and bunging it up a bit, increasing the activity. This little patch will keep going. Sometimes it splits, sometimes it dies off, but it keeps going forever. Just. There's a critical value at which it keeps going forever, And on this side, you're well in the turbulent regime. So when you look at the patches, there are lots and lots of them. They're completely splitting and merging, and new ones are appearing, and so on. And this will also go on forever.
What is the grayscale? Still vorticity?
The grayscale is that wherever you see something like this, you're seeing it's Enstrophy squared, but basically this is these turbulent patches. You're following the turbulent patches downwards. Yeah. Mm-hmm. That's right, a fluctuation will set it off. Well, it isn't, is it? It's active turbulence, but it means, yeah, if I did my simulation with absolutely no noise at all, I would not see this. This is with a small noise. Some activities, a small noise will do nothing. Here, it sets it off. But once it's set it off, we're asking really, does it go on forever or not? Yeah.
The first foundation creates a touch, then it appears at a planet. There will be no free-form breathing.
Well, there might be, yes. I mean, down here, there'll be another one, but they don't carry on forever. Yeah. Yeah. Yeah.
I'm just trying to connect this slide to the slide. So the white region, with periodic.
Yeah.
These are the interruptions.
Exactly. Yeah. Yeah, absolutely. Don't know. I mean, you were telling me about this. Yeah, I mean, it does look very much like other systems. I mean, I come back to that question, right? When we compare it. But I think the question you're asking about your inertial systems is very similar to the question which we can ask about these systems. Yeah. Oh my gosh, yes. Okay, we'll have a few questions and I'll finish the story and then have loads of questions. Yeah.
Intermittency, like this, or disappearance and disappearance and appearance.
You would, but you do need a tiny bit of noise to get it started.
But you see it, but there is still.
Yes, yeah. Yeah.
What's the mechanism through which the turbulent vortices sort of die off?
Dissipation, but that's a very sort of hand waving thing. And we don't know more than that, right? Yeah. This is along the channel. This is across the channel, and this is time. Yeah. So on the previous slide, this is across the channel, just a snapshot. All right. So I'd have one of those little black lines here and a black line there. And then I see how those evolve with time. But so I can put them all together. I've made it much thinner. Yeah. Yep. Yes. Sorry. Do you want to? Oh, you're just stretching, right? Okay, good. Is it good? Okay, so someone said directed percolation. This looks very like directed percolation. Directed percolation is a setup where you have a probability P that a site is occupied, or a probability P that a bond is occupied on a lattice. And then if things are occupied, you join them together. And on this side, you can see that there's a little cluster which dies out. And in general, if you have a low probability, eventually you never get a cluster which goes on from the beginning to the end. At the transition, you just get a cluster, which if you start here, you'll find a cluster connected at the end. And above the transition, you get a cluster which carries on all the way down. And you could do simulations of that, and we did, and we put them next to our active nematic, and you can see that they look very, very similar. And just to check that more carefully, what we did is we said, the turbulent fraction. So this is the amount of those black bits on average across the system. Okay, it's really saying, do I get to the end of the channel? Here you don't, there's a transition, and then the thing fills with turbulence. So no turbulence here except in tiny patches which don't span the system. Here you're just spanning the system, and there you're completely filling the channel with turbulence. You can measure the shape of the curve here. That shape of the curve gives you critical exponents. And for those of us that were brought up on physics 30 years ago, the universality of critical exponents is a very important thing, okay? The value of the exponents, the shape of the curve here is identical for our active turbulence and the directed percolation. They're in the same universality class. Somehow, that very simple model and the model we've got here, which is very complicated, when you actually ask about this transition, it's the same near that critical point. If you look at inertial turbulence, okay, so this is real turbulence, you know, you get the transition to real turbulence. If you look at the transition to real turbulence in a channel, what happens is that you can measure the critical exponent for that as well. And again, it's the same. It's within the directed percolation universality class. And you'll hear a lot more about this when you have the lectures in a couple of weeks' time. And I don't understand. I mean, we know about universality. We expect things to be universal, but it seems to me very strange still why this simple model and the active system and the inertial system are doing the same thing, even though the equations and the details are so different. Okay, so sorry, I don't know your name, but I know. So she's actually working on this, so knows much more about it than I do. So do you want to comment? Yes, they come out the same as well. Yeah.
Is it
Yeah.
2D or 3D?
This is 2D. These exponents are 2D exponents. Okay, yeah. I don't think so. Do you agree that it's not really understood? Go on then. Yeah. Yeah, I mean, they're working on it and doing well. So I think you'll hear more details about this. But as far as I know, it's not really understood. Yeah, yeah.
You have a little bit of deviation to that calculation because you have non-zero.
Not in an infinite system. But yes, we have, because of the fluctuation, you have to get it started somehow. In this case, it's noise fluctuations which get it started. My understanding is that in the low Reynolds number setup, you have to get it started by some sort of tiny perturbation or electro, I don't know how they do it exactly. But again, you need some sort of perturbation and noise on that thing. I think, that's right? Yeah.
Just a quick remark. This exponent beta is the same for shearing for canal system.
Oh, okay. Exactly the same, directed percolation. Okay, I've never seen that link. It's the same for predator-prey models as well. Yeah. Okay, so let's leave that and you can ask more expert people about that next time. And I'm really sorry not to be here for that. I maybe try and zoom in so that I can learn more about it as well. Right, so that was confined active systems. We're now going to do biology, and in particular mechanobiology, when physics is relevant in biological systems. So first of all, the disclaimer. It's not so different, I think, from these climate models, all right? That it's too hard for us to do the whole thing. So we have to try and pick out what's interesting. Where we're aiming is to understand systems like this. We can't do it yet, but this is what we're aiming for. This is Drosophila, a fruit fly. It's a baby embryo of a fruit fly, and it's growing into a fruit fly. What you're seeing here is cells. Each of these is an individual cell. This length scale is about 50 microns, and the time scale is about 2 hours. And this is the early life cycle of one of these Drosophila. Right, so this is an active system, right? It's taking energy, it's using chemical energy to move, to form patterns, and really to do very well organized changes to its shape. And it's pretty obvious that physics got something to do with it because there are flows going on in this system. We would like to understand things like that. This, I think, is one of the big questions at the forefront of mechanobiology. Nobody knows how this works. Yeah, it's got to decide, right? It's got to decide where its top is and where its bottom is. Oh, sorry, about the two things, all right. Looking at different directions. One's from above and one's from the side, yeah. Yeah. Okay, so that's the sort of problem we're going to do, but we would like to do. We're way away from that. So let's just start with some active matter. Oh dear, why is it doing this? Maybe it's when I hit the microphone.
Was there any kind of chemical gradient?
No, it's just growing up. I mean, they're probably internal chemical gradients. Internally, there's going to be chemistry. Yeah. I mean, if you're going to have an active system, you have to drive the activity somehow. You have to work out some sort of gradient in activity, presumably, to do something like that. 2 hours, about 2 hours. Yeah. Right. So I'm going to talk about two things which are way away from that, but on the beginning of a route to understanding what might be going on in these systems. And the first one I also want to do because I want to talk about three-dimensional active matter, because in the end, if you're going to do biology, biology is three-dimensional, not two-dimensional. So really, I'm going back now, forgetting about biology, to right at the end of this bit and talking about three-dimensional active nematics. In 3 dimensions, our topological defects are replaced by disclination lines. These lines form loops. They're allowed to form loops, but they're not allowed to start and end, or they're allowed to end on the edges of a system, on the surfaces of the system. There's a great deal of very nice geometry about these loops, but the way I sort of think it's easiest to think about them is to think about the cross section. So I've got a loop going up like this, all right, locally, and then it'll sort of loop round like that. And let's think about the cross section across these loops. You can either have the cross section looking like a -1/2 defect, okay? So the director field is in the plane and it has this threefold symmetry. Or if you look at another bit, what will happen is across another bit, it will look like a +1/2 defect. You call them a wedge type defect, so it's going to look like that. Or the new thing you can have is that if you look at a different cross section, what could happen was that the director actually points out of the cross-sectional plane. And that's called a twist type defect. And these plus and -1/2 are not topological defects anymore. They're not topological defects because you can change from one to the other by going via the third dimension. Okay. So this is now a movie of this microtubule motor system in 3 dimensions. Again, very beautiful experiments from Stonimir Dodgik's group, now in Santa Barbara. These green lines are these topological defects, these defect lines. these defect loops. And in order to see this, he had to take his microtubule motor mixture and actually put it in a background of colloids, a background of nematic colloids, because you needed this extra tendency to nematic order. Okay, so these are actual experiments looking at these things. And you can see the time evolution of these loops. Very clever experiments, but the physics actually is really pretty similar to the two-dimensional physics. Nothing very new that's happening, you find out. Okay, here's a loop. It's a loop looking at a close-up of a loop. And you can see loops can appear and they can disappear. But you can't have a line which starts and ends everywhere. And I think in a minute you'll see the directive field, which is really complicated in these 3D systems. There they go. Okay. So what we wanted to do, so we're back to simulations now, is think if I have a drop of active matter, what does it do? So it's a droplet, but it's not a drop of water, it's a drop of active matter. And so it's going to have these disclination lines in it. And the movie on the left shows the disclination lines. Sometimes you can see loops, but sometimes they just sort of ends on the edges of the drop, and that's fine, that's allowed. They're allowed to end on the edges of the drop. And the thing is vibrating up and down a bit because the activity is sort of pushing it. You've got forces there due to the activity and flows, but you've also got surface tension, which is holding it so that it's a proper drop. Now, it turns out that how these things behave is very different if they're extensile or contractile systems. And the reason for this is something called active anchoring. Okay, so what I'm going to do now is explain to you what active anchoring is. If I've got, a blob of liquid crystal, then sometimes you can get anchoring on the surface, that the thing prefers to have a certain, well, any surface with a liquid crystal, the liquid crystal will prefer to have a certain direction on the surface. So here are my surfaces, and if the directive field is like that, you call that parallel anchoring or planar anchoring. The way you make it happen is just by rubbing the thing. I don't believe this, but I mean, it's actually true that all they have to do is this, and then they end up with planar anchoring in the direction of the rubbing. Or you can have perpendicular anchoring or homogeneous, that one's homeotropic anchoring, and you do that by putting some sort of surfactant on the surface, right? So that's well known. In these simulations, we didn't put any anchoring on the surface. We didn't put any thermodynamic terms which prefers that or that. But you still get an effective anchoring due to the activity. The reason for that is that at the surface, you have a change in the order parameter, right? The order parameter has a value inside because you've got a nematic, and it has a value of 0 outside. And so there's a change in the magnitude of the order parameter between inside the drop and outside the drop. So we can say what we always say, change in the magnitude of the order parameter induces flows, induces forces, induces flows. So you get surface flows in these systems. And the surface flows line the director up like this. If you have get the right way around, extensile system, and they line the director up like that if you have a contractile system. So the contractile ones tend to look like that, and the extensile ones tend to look like that. Some of you may have seen this before because of how swimmers behave like near a surface. If you have a swimmer which is an extensile swimmer. That's a pusher swimmer. Have I got that one right way around? Pusher swimmer. They tend to like to line up and swim parallel to surfaces, again, because of their flow fields. And if you have a puller swimmer, they tend to end up want to line like that next to a surface, again, because of the way the flow fields interact with the surface. And I'm sure you'll hear about that in Tom Power's lectures in week 4. So you get a different sort of surface anchoring. The light brown one shows that you have an in-plane surface anchoring if you have an extensile drop. Have it about the right way around? Yeah. And you have this sort of contractile surface anchoring if you have a contractile drop. And this is the distribution of the angle of the director field near the surface. And really, those are just to show this is a large effect. It's a large effect. This effective anchoring is a large effect. And
It's held together by a membrane.
it's surface tension. So it's just like an ordinary drop. Surface tension, just surface tension, yeah, just surface tension.
And they like each other. They don't just.
Yeah, it's just like a water drop, except what's in there is an active fluid. Yeah,
like each
yeah, yeah. All right, so I'm going to show 2 examples of the dynamics of this drop, one for these extensile ones, which have pushes and in-plane anchoring, one for the contractile ones. And we'll see we get very similar things in real systems. So this is an extensile drop with in-plane anchoring. We've turned down the surface tension, so it's easier for it to deform. And what you can see is that it sort of sticks out these arms. Yep. They're really rather nasty fat arms, and that's just because of the resolution of the simulations. We'll see an experimental example where they're much thinner and much nicer. But we can look at the simulations and we can say, why is this happening? And the reason it's happening is that you get a line of essentially things which look like +1/2 defects across here. We know that +1/2 defects are self-propelled. They're busy being self-propelled in this direction, so they push this arm outwards. So the defects are pushing the arm outwards. I think this is really hard to see unless you've stared at it for ages, but just this is a line of defects like this. So what will happen is that the flows will push outwards like that. And then what happens is the defect line sort of escapes from the end and you lose the flows, and that's when the arms fall back again. This really happens in real systems. This is actually a very early experiment on actually a shell of active matter. So rather than a drop of active matter, it's a shell. And you'll see the arms appearing. These are much nicer arms. They're a much more sensible shape. And in the original paper, it says, and it looks like these arms come out from positions of the +1/2 defects. And this is one which nobody's really picked up on in the literature even since then, and really cute little thing, okay? And you can, yeah. The other nice one, which may or may not be relevant, is Hydra. Hydra is such a cute little animal. It's a tiny animal. It's a marine polyp. Okay, it's about a millimeter-ish in length. And what's cute about it is that you can cut a little bit of Hydra out. It's sort of hollow. It's a hollow thing. And you just cut out a little bit like that, maybe millimeter by millimeter. And you watch it and it grows. And it grows back into a Hydra. And it grows back into a Hydra, which has a mouth at the top, a foot at the bottom, and these 2 horns. And there's a beautiful paper from Kin and Eric Kerren's group, which links topological defects to the positions at which these arms grow. It's a bit different, of course. You know, topological defects in what? And the answer is in the actin cytoskeleton. Actin is a long filament, which basically makes the skeleton here. And it seems like it's defects in this actin skeleton which allow the arms to grow and somehow allow it to position the head and the foot. Just much slower length, slower time scale than we're sort of used to in the active systems, but similar physics perhaps. Yeah. Yes, you would have to have that to get the arms in the same place. It's not impossible because if you have a sphere, you have to have a +2 charge from topology. Often you will get that dividing up into 4 + a half defects, okay? And those 4 + a half defects tend on average to sit on the vertices of a tetrahedron because they tend to repel each other. This obviously is not exactly the same as that, but there are ways in which you can position defects due to elastic energy. But that's one of the reasons, you know, I think there's some question marks about, you know, this is not just a blob of active matter, it's much more complicated than that. Yeah. Only for the extensile case. I'll tell you in a minute what happens with the contractile case. I just play this, which is another simulation we did when we just looked at a shell, which actually has much nicer arms. Again, these form at the + a half defects. It's different from Hydra because in our simulations, the + a half defects escape from the ends of the arms and then they fall back down again. Hydra obviously doesn't do that. Maybe it I mean, one possibility is that it turns down its activity, but nobody knows. Okay. Yeah. That's not known. Hydra always, for almost, I mean, it doesn't always work, but when it works well, Hydra has 2 arms. Sometimes these things grow in the wrong way and it ends up with 3 arms or a foot and a mouth in the wrong place and things like that. But on the whole, it's pretty good at reproducing itself. Yeah. Okay, so that's extensile. Let's see what happens to a contractile one. I'll miss out this one, but I'm going to show you what happens to a tiny contractile one. Very different. This one is a contractile drop. It's a contractile drop. And so the anchoring of the pneumatic at the surface is perpendicular to the surface. And in this particular example, it's a tiny contractile drop. So it doesn't have room to form these defect lines. All you get is a single hedgehog-like defect in the middle. This single hedgehog-like defect is obviously really bad. Right? It's really bad because it has a huge energy. The thing doesn't like it. It certainly isn't going to stay like that. So what it does instead is it organizes itself so that it has the right anchoring here and here in the dark brown regions, but around the middle, it has a region where it has in-plane anchoring. That's just to reduce its elastic energy. Then what happens is that you have gradients in the order parameter, so you get flows. What the flows do for you is this. The flows organize themselves so they push the thing round and it invaginates to make a hole. Basically, it organizes itself into a hole like that. And the reason that's interesting is a thing called embryogenesis, which is the first stage in the development of most mammals. Right, so this is biology in a nutshell sort of thing, all right? We start lives as a I don't even know how to say it actually, one of those things, right? Which is basically a ball of cells with a gap in the middle, with water in the middle. Somehow you have to get from this thing, which is essentially is a one-dimensional ring of cells, to something which is 3-dimensional, so you can decide to be a heart or a lung or a liver or whatever. And the way that is done is by invagination. Basically, this thing here organizes itself. There are flows which push things in. And you end up with this thing which has one of these holes in, which has the shape that we found in the active system. Here's a picture of it happening in the chick. Bit hard to see because real movies in 3D are a bit hard to see. It's going to form down here. You can see lots of flows, and here it is pushing what's called a primitive streak inwards to form this dip. Yeah. Sorry, which one? Shout. Escape, you mean escape into the third dimension one? But essentially, that's what we were seeing yesterday in the bacteria, which were pushing upwards into the third dimension. Yeah. Here we don't, because we already are allowed that to happen. I mean, I've never done a cylinder actually. If we did do a cylinder, we'd certainly see it because that would happen, but I don't know how that links with the activity. Yeah. Yeah. Why do you think these sharp features? Well, I mean, this is one of the problems. This isn't that sharp, right? It would be nice if and the problem is that we have an interface width in these simulations. So that screws us up. Our arms are far too wide compared to reality. Yeah. Okay, with surface, no, that's a very good question. In biological systems, you have very low surface tension. You stabilize things with curvature energy. And one of the things we want to do is see how much it makes a difference if we have curvature energy here rather than surface tension. But numerically, that's much harder because you're a higher order in derivatives. Yeah, yeah. No, no one's seen anything to do with defects there. I think that's a very different system. Yeah, I'll say anything about that soon. Yeah, okay. So the biological systems are very different to the simulations, but we just did the simulations and we immediately got this gastrulation, this invagination. It's surprising there's not some sort of connection between them, but there's an awful long way to go before we can take a ball of active matter and say that it's anything like reality. I mean, this is very much the current direction of research. So what we'd have to do next is actually stop worrying about continuum active matter and instead look at cells, real biological cells. Things which are squishy, things which move like this. This is a picture of MDCK cells plated on a surface. And if you look at them, they do this. So we have to sort of get a bit further down towards what's going on. You know, in a continuum picture, that looks like active turbulence. It's actually starting to go round and round. That's because it's active turbulence in a box. So just like in a channel, things start moving. Here they start this coherent motion. It's active turbulence, right? Again, but these things aren't nematics, right? Then they're on average, they're isotropic. I was going to say circular, but isotropic. If you averaged over them, they'd be isotropic. But you are getting active turbulence like things, okay? So we've got two reasons to study these. First of all, it's another example of active turbulence. Is it the same as the sort we had before? And second, if we are going to claim that these active systems are anything to do with biology, we've got to start somewhere. And where you're going to start, you're going to start with these cells. The white spots is, no, that's where the cells are dying and getting out. I mean, at the moment, it's not obvious there are topological defects in there, right? I mean, they're going to turn out that there are, but it's not obvious at the moment. And I should also say that where we are at the moment is we're tending to look at these two-dimensional plated cells, right? So it's cells actually sitting in 2 dimensions. In reality, cells are in 3 dimensions, but we've got to start somewhere. So what we're looking at, mostly what almost everybody is looking at, is these epithelial cells. Epithelial cells do tend to form 2-dimensional layers, okay? They can look like this and like that. And they're the ones which line the body. So it lines all the body cavities, its skin, it sort of is a protective coating and a membrane where transport goes across between different bits of the body. So the question I want to ask is, how do these cells move? I mean, can I write down a simple physical model of what's going on there? So where would we start? Well, okay, so we'd start with a single cell. Can we understand how a single cell moves? Well, the biology is that you get these actin filaments, which are long and rather stiff polymers, which push the front of the cell forwards. The cell grabs hold of the substrate due to focal adhesions and sort of pulls itself along. Okay, so it's not swimming, it's not like a swimmer, it actually has an external force on it because it's pulling on the surface. And then because it gets a bit elongated when it does that, there's some machinery here which pulls in its tail. It's a load of motors which sort of contract a network of actin here, which helps pull in the tail. So if I want to be a physicist, all right, how do I model that? Well, I model this as a force which is pulling the cell forwards, a polar force, i.e. one that's got a certain direction. So there's a force pulling this cell forwards. And then you have this contractile force, which is tending to restore it to circular, which is an internal force. It itself has this machinery to pull itself back to circular. And it's a contractile force because it's shrinking the cell along its long axis and as a result, pushing it outwards along its short axis. And that actually works very well, just to sort of these things, if you have a persistent polar force, if the polar force is linked to the velocity, you can work out the direction a cell moves. It's a persistent random walk. And for many cells, that works really well. I think I better show you a movie of a real cell moving around just to make clear this idea that physicists don't get it all right. And it's a nice movie. The reason this movie was made is just to show how much better, this is the old version of the same thing, really to show how much better they can do the imaging now. Okay, cells actually move in the body through the extracellular matrix, which is a great mass of fibers. So this is much more like reality. Yeah, and these are neutrophils, which are cells which move very fast. Okay, so that's how a single cell moves. Yeah. A normal cell velocity would be a micron per second, I think. I'm wondering if does anyone know? Micron per minute, maybe. Oh, no, I'm sure they're not that slow. And then they are very slow compared to bacteria and things. No, it's not, no, because we better look it up on Wiki, sorry. My guess is it's a micron per second, but maybe we're right in the middle, sorry. Okay, so now let's ask, how do layers of cells move? So these are it's a bit special because this is an experimental system, it's a model system, and the layers are plated on the surface, so you have a two-dimensional layer of cells. Sometimes they can just get stuck, you can get them jammed. So basically you've got this layer of cells, here are the boundaries between the cells. It's called a confluent layer because they're hanging onto each other hard and there are no spaces between them. Okay, sometimes they'll wriggle about because they're active and they never completely stop. They'll wriggle about, but they won't move relative to each other. That's a jammed state. They can also flock. Okay, so this is a real movie of real cells moving. Flocking means they're all moving in the same direction. For example, this is the egg chamber of this Drosophila. It has a layer of cells on its surface and the cells move round like this. These are experimental pictures as well about these cells moving together in the same direction. So this is almost a bit like that sort of active flow down a channel. So they can flock. And the other thing they can do is active turbulence. We saw the picture, the movie. I'll show you again. This looks like active turbulence, at least to start with, it looks like. If you look at the velocity fields, one is the cell velocity, the velocity of the different cells. One is active turbulence. You see the same sort of vorticity. You see the same sort of velocity jets. If you put a load of cells in a channel, they will start moving just like active material. These are lovely recent experiments. What happens is the cells set up a shear flow within the channel, and you can do active pneumatic theory and you can match it exactly to what's happening in that channel. And the other thing you see is topological defects in these systems. So how on earth can you see topological defects in cells which are isotropic? Okay, and this is a mapping which shows these cells are isotropic, but on here we know, right? So I think you're right, microns per hour. Okay, and the time scale. All right, so that gives us the time scale. These things are moving a few cell lengths in that time. Okay, so what happens locally is that the cells pull on each other, and so a cell will be pulled out like this, so it's elongated. So it's transiently pneumatic, where transient means an hour or so. And then it's going to pull its neighbors, and it's going to pull them in the same way. And so you get fluctuations, you get pneumatic fluctuations in this system. The reason you can get pneumatic fluctuations is that they're squishy particles. So if you pull one, you pull the others. They're particles that you can deform. And so you can stick a pneumatic order parameter on this system by taking the average elongation. And you can see it sort of makes sense, right? Because the pneumatic here, you get this pneumatic ordering. It doesn't last everywhere in the system, but you get locally, you get pneumatic ordering. And then you can plot the pneumatic director that you get from your cells, and you can find topological defects in here. And these topological defects move. Okay. And I mean, this was the first observation of these things. It's a paper we wrote and a paper we were very surprised to find these. People didn't believe them for ages, but now they've been found in loads and loads of different cellular systems. Okay, so you're getting defects. They're extensile defects, and that's a bit weird, isn't it? Because we said that cells are naturally contractile. We didn't understand that for a long time. And indeed, if you think about it, if you've got cells which are extensile, it means that they'll tend to be more pneumatic, because if they start pneumatic, they'll end up pulling themselves like that. Whereas contractile cells shouldn't work, right? Because if they're pneumatic, they just go back to being isotropic. So that's the question we're asking. We understand there's different ways in which these cells move. sorry, that's quite the wrong to say. These are the different ways in which the cells move. And these are the questions we're asking. How on earth can we write down a simple model which gives us flocking and jamming and active turbulence, a simple model in terms of the forces between these cells? Okay, so the model that people use to look at this sort of thing, it's no use using a continuum model, at least to start with, because you have to understand the underlying physics that has to be put into your continuum model. So people use these things called phase field models. So what these are is a load of squishy cells joined to each other. All right, so think of bubbles. I don't want to go into this in any detail. Think of bubbles, all right? If it was a passive system, it would be a load of bubbles, but the extra thing you have to do is force it to make it an active system. And if you put on forces, you can get it moving around, and it's starting to look like cells would move. So there you have a load of cells sitting there, moving around. How do we put on the active forces? That's really the question we're asking, right? How do we add the active forces? What's my simplest physical model which can give us the right answer? Well, we know how single cells move. Basically, you get this polar driving, you get a force on each cell, which tends to link to the velocity. Let's just put that on. Let's just take all those cells in my phase field model and just put a force on each of the cells and allow the force to relax to the velocity, because it makes sense that it just makes physical sense to do that. And if you do that, what you end up with is almost the right answer. If the force isn't very strong, the cells stay where they are. These are the center of mass of the cells. So you have a jam state. If you increase the force, you get a liquid-like state. And if you have a strong link to the velocity, whoops, you can get flocking. You can see these things are going to end up moving around, and then they're going to start flocking and move in the right direction. So we thought, great, this is great. We've got everything we want. We've got a liquid-like state. We've got flocking. We've got a jam state. Let's look for the topological defects in the liquid-like state and prove that it's like active turbulence. No topological defects, completely the wrong correlation function, perfectly normal liquid, absolutely nothing like active turbulence, doesn't work. So putting just a force on each cell is wrong. It's just not giving us the right things. And we sort of should have known that, because there's something called contact inhibition of locomotion, which means different things to different people, but for us means that if you put cells in a layer and join them to each other, they stop making lamellopodia and they stop doing these forces. They stop making these polar forces. And that makes sense, right? Because if you're a cell, if you're making a polar force and your neighbor's doing one in the opposite direction, you're just going to all be pulling against each other, and that's a complete waste of energy. So it sort of makes sense that they actually stop bothering to try and move once they're in a monolayer. And this is actually what happens. Nobody understands why. It's one of these biological beliefs, but it makes a lot of sense. And to see what they might do instead, they're absolutely beautiful experiments from the group in, where are they? Group in Italy. And what they did is that they had a load of cells which were doing active turbulence. So they were happily zooming around in a slightly random active turbulent-like state. And they added RAB5A. It's a chemical which is meant to destroy cell junctions. And what happens, it doesn't completely destroy them, but it destroys the signaling across them. So the cells still stick together, but for some reason they don't seem to be able to communicate in the same way. No one's quite sure how. And what happens is that these things start flocking. This blue line is the velocity in active turbulence, the mean velocity. You add this chemical, and the cells all take off in the same direction. Let's look at a movie of them doing that. On the left, we have active turbulence. On the right is what happens when you add the chemical and the cells all take off. Okay? So what this is saying is that cell-cell junctions are important. It makes a difference if you muck them up. And actually that makes loads of sense, doesn't it? Okay, yes?
They seem to slow down at the end of the movie.
That's because they're going to run out of food. Yeah. Okay, so we want the simplest way of modeling the dynamics of this cell layer. We don't have a polar force. We don't have a monopole force. So what's next? The dipolar force. Where's that going to come from? Well, it's going to come from the cells pulling on each other, right? When you've got loads of cells, they're all active, so they have to be moving. They're going to pull on each other, and pulling on each other means equal and opposite forces across the cell boundaries. And that's exactly what we mean when we talk about this pneumatic-like dipolar force, equal and opposite forces again. And that makes a lot of sense. It took us ages to realize it, but I think it does make a lot of sense. You turn off the polar forces. These things are active. They're cells, they're active, right? So they're always moving, even if they're not got any net direction. Contractile forces is what they do, because they're contractile cells. That's what they're set up to do. And intercellular interactions are likely to be important in this system. Intercellular interactions are equal and opposite. And so we know how to do equal and opposite forces. It's this minus zeta q thing. Let's put that into the model and see if it works. So we put it into the model and we ended up exactly with what we wanted. We ended up with active turbulence. Okay, it's very slow active turbulence because they're cells and because it's really hard to do these simulations for long, but you get these topological defects and you get the sort of splay instabilities that you get in active turbulence.
How's the dynamics that's different from 2 lectures ago?
The dynamics is exactly the same. I'm sorry, it looks different because we're doing a different model. We're much slower, but the dynamics is exact. Well, to a first approximation is exactly the same. It's an active contractile system. Somehow, the cells are being stretched by contractile interactions. So pretty much the last thing I'm going to say, because we're all exhausted, right, is why on earth can contractile interactions, which are meant to do that, why on earth can those extend the cells? They can look like a pneumatic and do active turbulence. And the answer is because it's contractile interactions between cells. Here's my test cell, and here's a cell next to it. This is its long axis. This cell's being contractile. And so what it's doing is it's pushing like that on the test cell. The cell down here is being contractile. And so it's pushing like that on my test cell. Cell out here is being contractile. And so it's pulling like that on my test cell. Similarly on that side. Of course, you have a different number of neighbors, but I'm making it easy. Okay, so this one is actually extended. There's an effective extensile interaction because its neighbors are trying to contract. And that's why it works. And so we now believe that, at least to a first approximation, and this is, I believe, not the rest of the world believes at this stage, okay? But I think it's an interesting way to go down, and it sort of works, that contractile interactions between cells, the most obvious thing you could have when they're pulling on each other, will give you this contractile active turbulence. And I'll just say it because it's late and it gets very complicated. If I then add fluctuations in the size of the cells, okay? So these things are never this simple, you know, we saw this fluctuating all over the place. What happens is that that actually changes that to an extensile system. No, it's not obvious, but that's another story. Okay. So what you can see is a huge gap, right, between what we can do. We can sort of starting to understand cell layers, but only as a physicist. Life's really much more complicated. There's the underlying biology, which says, how do these things pull on each other? In 3D, we're seeing things like the biology, but the biology is much more complicated. You know, where does the activity come from? There's underlying chemistry, there's time dependence. Lots to do, but it's fun. So that's what we've done, got through a lot. Thank you very much for listening. Enjoy the rest of the thing and nice to talk to you.
A few more minutes for questions.
Yeah. Yes. That's a good question. I think it's going to depend on the amount of noise you put in, right? You're going to get more patches created if you have more noise. And then they're going to keep going if the activity is high enough and you're going to transition to turbulent. So I think it just will be a noise effect. Yeah.
Professor, I just have a wild imagination. I'm wondering whether this cell interaction can be compared to the really beta cell, because in really beta connection, I also see, you know, each convective cell, when it expands in one direction, for example, it squeezes its neighbor and therefore influence its neighboring convective cell. So if there is a background flow or something, I also observe something like, you know, the shift of the pattern. So this is just something inspired by your talk.
Maybe that's what we're here for, to look across different systems. Yeah. No. There are definitely more. People have looked at a shell of active matter and they do see that it settles down to having 4 plus a halves or 2 plus 1s. The topology here is a bit different because we haven't got a spherical shell, we've got the whole thing. So we're not point defects, we're lines. So it's ends of line, which are a bit different. Yeah. No.
Correlation like scale to the size droplet, or like that, if that's similar.
The scale you're looking for is this active length scale root K over activity. That tells me a typical spacing between these defect lines, and you'd need to compare that with the scale of the drop. So the first one I showed, you could fit defect lines in, so it was smallish compared to the scale of the drop. This one where we got this invagination, you couldn't, and so the active length scale was large compared to the shape of the size of the drop. Yeah.
You said in the contract that is basically like in the perpendicular.
Yes.
Very small drop.
The reason is that gets a bit of a list if I talk about small ones and big ones. But what the big ones do is they end up looking like walnuts. I'll show you the movie. I mean, I was talking about that because it's the invagination, which is rather fun from a biological point of view. But this one is the back one. Is that going to work? Yeah. This is large contractile drops. What happens is you get defects pairing up on the surface and you get flows which give you something which looks like a walnut. These are stills of it. And again, it's the flows which are giving you this sort of shape. Yeah. Yeah. I'm not sure I would know what they were, but the only time we see something funny happening on the surface is where a line, a defect line hits the surface. So what would a boojum be? And then you have a defect line like that. Yeah, you get those. Yeah, talk to Marana. She's an expert on that. Yeah.
The flow profiles that you showed is quite geometric. It looks to me very similar to the one that was observed in active rollers.
Yep. It's very similar. It's if you have a confined system and it's active, you'll get this. Yeah. And then if you then bump out the activity again, you tend to get these sort of bits coming in because you're getting an instability. Yeah. Yeah.
You introduced this. And I was wondering, making the connection between observations and theory, quantify this activity. Do you use the scale of the structures to observe?
It's hard. People are doing machine learning when they look at the experiments and try and match it onto the continuum equations. And that's probably the best way people have done it. And yes, the active length scale will give you one idea, but then you have to know K, which is the elastic constant. So people are trying to match, because it certainly isn't obvious that these microtubules, which are wriggly, are exactly the same as a normal active, an active pneumatic, which is pneumatogens which don't bend. We saw an interesting poster on that last night. Yeah. Yeah. If you're talking about our droplets, no, it's just an ordinary drop and it's kept there by surface tension. In the experiments that we saw with those lovely thin things, there would be a curvature energy, I think.
The energy cost due to the curvature is increased. Pressure in that curvature region, so energy of the membrane as a whole will increase. So how this system is it whole system, the energy of the whole system will increase, or it's like somehow it's stabilized with different.
It will increase, but that's fine because you've got active forces acting.
I mean, in time, if I track the energy, will
It will increase, but it won't increase to infinity because then, you know, you lose the defects and then it comes down again. Yeah. And you've got velocities in there as well. So that's the force which is doing it for you. Yeah. Yeah. No, this comes out from the flows. What we actually put in our simulations is no thermodynamic anchoring. Yeah. Because it's active, you get this effective anchoring. And the reason you get that is the activity induces flows which move in such a way to give you the anchoring, to push the pneumatic director around. Yeah. It depends whether it's an extensile drop or a contractile drop. But remember, these are simulations, so I can muck around with things as I like. Yeah. In the experiments, an extensile drop would have extensile planar anchoring, and a contractile drop would have perpendicular anchoring. You can see this in bacteria, actually, little drop of growing bacteria. Growing bacteria is like extensile activity, and they always align parallel to the surface of the drop. Yes?
So I have a question for the first part of active surface in chemical. So you mentioned 3 states for laminar flow, oscillatory flow, and then bouncing. And I'm wondering, in the oscillatory flow, the cell will reverse their direction similar to the bouncing state, or it will take the same direction but oscillate?
It's the same direction. So basically, it's sort of just about trying to have an instability which then dies out. So it's a state with a net flow. So if you're doing microfluidics, that's what you want, that sort of state. Yeah. Yes. The phase field simulation, yeah. So we have a variable which is 1 inside a cell and 0 outside. Same for all the cells. Yeah. So they're like droplets with an edge to them. And then we put in things which make them look like cells. For example, an adhesion energy, so they stick together, but an energy so they don't overlap. And then a Landau energy, which means that we have a sort of sensible interface between inside and outside. So it really is like a load of 2D bubbles stuck together if it was just passive. And then that relaxes to the minimum of the free energy. And then you add these active forces. They're called phase field models, and they're pretty well established, but yeah. Nice review by Ricard Alert, A-L-E-R-T, on that. Yeah.
We'll let people head off to lunch. So 2 p.m., what have we learned session? I think Leo is suggesting we'll try to get them organized with some lectures up here, give 4 or 5 minutes. Notions of
Oh, no more.
nobody's forcing you to do it. It's artistic